So hi, my name is Appanna. I'm working here in the Appanna Lab. Previously, I did my undergraduate degree in biochemistry here at Laurentian University. And actually, my current research right now is looking at dormancy in cells, so looking at after cells go past their stationary phase, how they're able to survive, a bit in terms of the metabolism, and then how these kind of contribute to things like antibiotic resistance and super-resistant bacteria for diseases. So the goal of my research is kind of to look at these dormant cells and sort of figure out a way to sort of affect their metabolism in a way that won't affect humans, so that you can kind of kill them or shut them off before they start reproducing too much. Because in that way we can stop a lot of bacterial infections, particularly with antibiotic resistant bacteria. Right now, a lot of the antibiotics we use are starting to become less and less effective against treating bacterial infections. This is because after multiple generations, they've kind of figured out a way to circumvent the use of these antibiotics. So we want to try to find a different way to deal with a lot of the diseases that these bacteria cause. And I guess looking at it from via metabolism would be one of the best ways to do it. So dormant cell is pretty much a bacteria that has gone past the stationary phase. So usually when you're looking at bacteria in their normal growth phase, past stationary phase is usually when they don't have enough nutrients or a carbon source to continue surviving. The idea of that is that most bacteria will probably die, if not all. But there is a small percentage of these bacteria that will survive, and they'll survive in a fairly small amount. One of the principles is that when a new nutrient source is added, these bacteria that have somehow survived in this very harsh environment will start reproducing again, and these new dormant bacteria will be a little bit more resistant because they were able to survive in these conditions. So I kind of want to look at antibiotic resistance in this because my previous undergrad work was looking at bacteria and how they kind of adapt to various kind of stressed environments, and kind of looking at antibiotic resistance, and specifically in dormant cells, kind of like that natural next step. So yeah, bacteria is quite important, quite important area to study because they cause a lot of infections, particularly now with superbugs that are in the hospital. It's important to look at bacteria in that sense because we don't want these to get outside or to continue procreating in the way that they are. So looking at bacteria, and particularly the way they're able to adapt to things like antibiotics and other stress environments, is very important in dealing with infection and treating diseases. In terms of bacteria and their metabolism, it's quite different than humans because, as we've seen, especially with the bacteria that we use, P. fluorescens, their metabolism is quite versatile and hardy. So even stressors in the solution that would normally affect other animals like us quite heavily, they've found ways around it so they can continue to survive and be viable. Bacteria are more adaptable than we are. I mean, since we're a multicellular organism, we have numerous moving parts in our bodies, we can't regulate as much. We want a bacteria, it's just the one cell, which means that it has to change the one metabolism, which is often why it's able to adapt to these various harsh environments to continue surviving. By stressed environments, I usually mean just that there are not enough nutrients for a normal bacteria to survive in, so they would have to figure out a different way to continue surviving with limited nutrient source. Metabolism is, the way if we look at humans, whenever we eat food, how it's broken down, and then how it produces, energy for us to move our arms, to think, to see. All right, so in terms of my methodology, you start off with step one, which is choosing the bacteria. We've chosen P. fluorescens, which is a fairly versatile microbe. We've used it a lot in this lab. Basically, it can adapt to different environments, depending on what kind of stress, what kind of food source you put in there. So that's why we've chosen it. Step two is that we grow the bacteria in a nutrient broth, which is just a food source for the bacteria to grow in and proliferate, so we can get up a fairly good number of bacteria. Step three is separation of your soluble portion, which is kind of liquid part, and the more solid portion, which is your protein. The liquid part, you can do an analysis on it for metabolites, which are the things that are in your metabolism, the kind of the various compounds you find there, and looking at what they kind of tell us about the enzymes that are being used. And that solid part kind of goes into step four, which is looking at the enzyme activity that's occurring inside each of the bacteria. So usually we deal with normal and a stressed bacteria, and then we got to look at kind of how the metabolism in each are different based on the kind of enzyme activity we see in each one. Well, ideally, if we want to try to affect the metabolism of these creatures, we want to try to have it maybe at least 24 hours is at the minimum. So we want to try to kind of affect them in that kind of extremely fast time. Ideally, we want to try to affect it before reproduction occurs, just because at reproduction you're dealing with way more cells and a way more complex system. Well, there are a lot of challenges when dealing, especially in a science lab. You know, you got to look for funding, but also things that are more practical, like time and how long it's going to take, and also, you know, figuring out the best method to do something, because you may be able to find something in a paper that looks like a good method, but it may be too time-consuming, you not have the material, or that it just may be a better, more efficient way of doing things. So, just some of the few challenges that you face when working in a science lab.